Good morning, everyone, and thank you for attending this symposium. My name is Sagar Dagani, and I'm an associate professor of medicine at Mayo Clinic in Rochester, and also the chair of the epidemiology subcommittee for the ADA. Today, we have a wonderful symposium scheduled for you guys, and we have some outstanding speakers from the University of Alabama, Indiana University, Arizona State University, and closer to home, the University of Chicago. So as we do that, as each speaker comes up, we'll introduce them and they will take you through their presentation. We are right on time to get started. Our first speaker is Dr. Nicole Armstrong from the University of Alabama at Birmingham. She is an assistant professor and she is going to talk about heritability of type 2 diabetes across diverse populations. Dr. Nicole Armstrong.
Thank you so much. Guess you guys can hear me. I speak kind of loud, so shouldn't be too much of a problem. Good morning, everyone. Welcome. So this is a genetics and epidemiology lecture or symposium really, first thing in the morning. So thank you all for coming at 8 a.m. on a Saturday. I am Nikki Armstrong. I'm an assistant professor, as just mentioned, at the University of Alabama at Birmingham. Let me see if I can. So it's the clicker. Got it. So I have nothing to disclose, and feel free to take pictures if you feel like you want to. No pressure, either way. So just a quick overview. The first thing I wanted to spend a little bit of time is what is heritability? Why should we talk about it? What is it and what is it not? How do we estimate it? So things like what are some tools that we do? What are some measurements? Why ancestry and diversity matter in this space. And then I want to present some results from a larger scale heritability analysis in Black adults. And then we'll talk a little bit about some of the implications and where we go from here. So I'm not going to spend too much time here on this slide. You guys all know these trends better than I do. But it is worth grounding our conversation that type 2 diabetes continues to grow in prevalence and impact, both globally as well as in the United States. So using data from NHANES, these two graphs show significant rise in the diagnosis really of diabetes, type 2 diabetes, especially among men. But let me get that. You guys can't see my mouse. But undiagnosed rates really haven't changed too much across time. So what is important and relevant to today's talk is that risk is not equally distributed. So age-adjusted prevalence varies sharply by race as well as sex, with non-Hispanic, Black, and Hispanic individuals experiencing some of the highest prevalence rates. So some groups do develop T2D earlier, it progresses faster, or they can experience worse complications. So understanding genetic contribution to that type of variation is a key step in moving towards more of a equitable and precise precision and care. Okay, so that's kind of where the genetics part comes in. Okay, so most of us are trained to think that T2D is a lifestyle disease, and it absolutely is true. So diet, exercise, social determinants of health, they all play a very important role. But we also know that people respond differently to these different types of exposures. So one person can eat, a high sugar diet, and their glucose stays within a normal range, while another can develop diabetes relatively quickly. So that variation often reflects underlying genetics. So heritability and heritability estimates help us quantify that. So it's not about predestination, it's about how much of the variation in disease risk across a specific population can be explained by inherited genetic factors. So this can help clinicians think more critically about family history, things like genetic testing, risk stratification, and it tells us as researchers what proportion of the disease liability we might uncover really through genetic studies. So heritability estimates are really giving you the ceiling estimate for things like polygenic risk scores, like how much can we actually get through these downstream tools. So what is heritability? Heritability is the statistical estimate of how much a genetic difference can explain disease risk across people in a population. Okay, so it is a population measure, not an individual risk. What it is not is like how genetic a specific disease is. So one way to think about this is if we could hold everyone's environment constant, same diet, same activity, social stressors, the heritability tells us how much the genes would account for variation in who gets T2D. But it does vary. So if you estimate heritability in a very homogeneous population, it could look high because everyone has similar environmental stressors or factors. So when we say heritability, we're often, at least in the gen epi field, referring to narrow sense heritability. So you can see that denoted as a lowercase h squared. So this reflects additive genetic effects, or basically how much a single allele will add to the disease risk independently. So there's also broad sense heritability, and that's the capital H squared. And this includes all genetic influences. So additive effects, dominance effects, and epistasis. And those are listed there if you're curious. But these are typically more family-based studies. So twin studies, these tend to estimate these broad sense heritability measures, which is often why the numbers or heritability estimates are a little bit higher than the narrow sense estimates. Okay, so SNP-based methods, which you can get through observational studies, it could be sequencing, it could be array platforms, estimate this narrow sense heritability, and that's what I'm going to talk a little bit more about today. And they capture the additive effects of common variants. So those variants with an allele frequency essentially greater than 5%. So relatively common in different populations. I hit the button. And so there are two primary ways we estimate heritability. We talked about this a little bit, broad sense and narrow sense. So family-based methods, which are over here on the left, they estimate, again, this broad sense heritability. So it captures all sources of genetic variation. But they also include some shared environmental effects as well, which can inflate estimates. So SNP-based methods like GREML, which is genome-based restricted maximum likelihood, which we'll talk about here in a couple slides. These methods, or like LD score regression is another one, these just estimate the additive effects of these common SNPs. So SNPs are these genetic variants. These estimates, again, are often lower because genetics, you're only seeing a subset of the genetic component. It's not that the genetics contribute less. So both methods offer very valuable insights, but it is kind of important to note it's kind of doing a disservice to both methods if you kind of compare them to each other, because they are asking slightly different questions. Okay. All right. So one of the more important points in the talk today, so if you get anything, it's mostly kind of this slide. I don't care if you know the methods so much about heritability, but heritability is population specific. Okay. I don't know how many people do genetic studies, but most genome-wide association studies, or GWAS studies to date, have been conducted in European ancestry populations. So when we use that data to estimate heritability, or do things like polygenic risk scores, which we're trying to apply, clinically, or you do searches for all of these different causal variants, we're doing so through a very Eurocentric lens. Different populations have different allele frequencies. They have different leakage disequilibrium, or LD structures. So a risk variant that is common in European ancestry might be rare or absent in individuals of African, Asian, or an admixed population. So likewise, environmental exposures and interactions with genes differ. So that's what this image is looking at. So in 2019, there was this big study published in Cell that was looking at all of the individuals with genetic data published through these large studies, and nearly 80% of those individuals in these studies were of European ancestry. This is important because this is kind of the basis of all downstream, all of these risk prediction tools, so on and so forth. It comes from these studies, kind of right here at the beginning. So if you're trying to build inclusive genetic research, scientifically rigorous research, we want to make sure our tools work well in all patients. And in order to do that, we need to focus and develop these tools with all patients in mind, right? Okay. So a little bit about the actual study that we published. We were focused on building the need for population-specific heritability estimates. So just due to the data that we had accessible and available at UAB, we set out specifically to estimate narrow sense heritability for T2D in African Americans or Black individuals. We used 3 large cohorts, so Regards, GenHat, and Emerge, and I'll talk a little bit about that here in a minute. And then 2 complementary methods, okay, GCTA and LDAC. So GCTA is widely used, has historically been a gold standard heritability estimate, but it can overestimate heritability, specifically in areas of high LD regions. So basically they don't work as well in non-European populations. LDAC incorporates LD as well as how rare an allele is, or minor allele frequency, in the SNP weighting. Okay, and that's especially relevant because there's greater haplotype diversity in African ancestry genomes. So if you think about those genomes, they've typically have shorter haplotype blocks, just based on evolution, them being, the genome being around, essentially longer being the common ancestor. So it's chopped up into smaller blocks. So they have different patterns there. So in both methods, both LDAC and GCTA, we utilize a common overlapping set across all 3 cohorts of about 8.5 million imputed variants. And then the T2D case definitions were all a little bit different. So in Regards, and again, I'll talk about the studies here in a second, but T2D was defined as a fasting glucose of 126 milligrams per deciliter, or non-fasting glucose of 200 milligrams per deciliter, or the use of glucose-lowering medication. And that was based on medication review and self-report. In GenHat, so GenHat is an ancillary pharmacogenomic study to a clinical trial, AllHat, which is an antihypertensive trial. Based on criteria, the fasting glucose cutoff was 140 milligrams per deciliter. That's how they define diabetes in this, this trial. So we used that, or the use of glucose-lowering medication. And then Emerge, we used validated methodology with ICD codes. And then the cases and controls, we tried to keep it at a relatively equal case control ratio, and there were about 10,000 or so individuals there. 15,000, sorry. In the next slide. So the 3 cohorts provide a unique opportunity. So Regards is population-based. It's longitudinal data. It was originally a study looking at stroke risk factors, racial and geographical differences for stroke, but it also includes a lot of information on cardiovascular and cardiometabolic diseases. GenHat, again, it was derived. It's a pharmacogenomic ancillary study to AllHat. So these individuals had clinically adjudicated outcomes, and a lot of the individuals were diagnosed with T2D because it was a well-established secondary cardiovascular outcome. And then Emerge is based on EMR or EHR data. So despite these differences, we harmonized the phenotypes as well as we could. The differences are a little bit one of the limitations. All of the genetic QC was harmonized to create this analytic sample. And I'm not going to go through the table, but basically it summarizes key demographic differences. There were 15,000 Black American participants, a relatively balanced case control cohort, or case control counts across all cohorts. Average age, so the mean age was similar, although the age range did differ a little bit. And apparently the mouse works to flip. So we're going to go just to the heritability estimates of T2D. So this summarizes basically the entire paper in one kind of simplified table here. But again, these are SNP-based heritability estimates for all 3 cohorts independently, and we use LDAC and GCTA methods. So we generated these methods based on an observed scale, so basically what the results are. And then we also looked at them on a liability scale. So liability scale uses population prevalence of the disease in the measurement. So it takes into account. And we use the population prevalence of 12.5% based on a published paper. And this is, again, African ancestry specific. So just a few key points to highlight. Across all cohorts, estimates in LDAC tended to be slightly lower than GCTA on both scales, although patterns are consistent. Regard, specifically, LDAC estimate liability scale was 22% compared to 24 and 29% from GCTA. And then GenHat shows the lowest heritability across methods, and eMERGE show the highest heritability across methods, and we're just looking right in here. So one of the reasons, for this, we kind of estimated it probably reflects differences in both the sample ascertainment and the different methods we did. So EHR versus a study population, a national representative population, and then a clinical trial where they were looking at individuals with cardiovascular comorbidities. It could also reflect differences in population structure, the actual phenotype, so T2D differences throughout all of those different cohorts. So the main takeaway here is that these findings reinforce that heritability is highly context specific, which I kind of talked about a little bit before, not only to ancestry, but also to study design, case definition, and population characteristics. Okay, so what are some implications? That's pretty much kind of the take home here. Our study confirms that a substantial portion of T2D risk in African Black Americans is due to common additive genetic variation. Estimates range anywhere between 18 and 34%, which is fairly common to what you see in twin studies. These results are modest compared to those that we have observed in European studies. So it really underscores two important points. First, that common SNP variation does contribute meaningfully. So using things like microarray data or these SNP chips can be helpful. Second, underrepresentation of these populations, specifically in this case, African American or Black Americans, and genomic and genetic studies in general could directly limit access to future tools based on this information. So again, I keep mentioning polygenic risk because I think there's a whole session later today or tomorrow on polygenic risk scores. I think it's tomorrow. As well as prevention. So our study helps close this crucial gap by generating these heritability estimates. So where do we go from here? I'm going to wrap up really quickly. We need larger genetic studies, GWAS specifically, that specifically prioritize inclusion of other ancestral populations. So there are plenty of undiscovered loci, very many, to type 2 diabetes that we have not identified, and that will ultimately lead to the heritability in these groups. So we also need to incorporate things like rare variants and structural variants. Those were not fully assessed here. So we just did very common genetic variation. From a methodological standpoint, reference panel harmonization, ensuring consistent definitions across type 2 diabetes, like whether it's a clinical trial, whether it's a longitudinal study, observational study, community-based settings, EHR data, it needs to be critical. And ultimately, our goal is to ensure that insights from genomic research actually translate to prediction and treatment tools that are accurate across all populations, not just those of European descent. Otherwise, again, kind of the take-home message, precision medicine may inadvertently deepen what we already have with existing health disparities, especially in this area. So with that, I just want to say thank you to some of the collaborators at UAB, both the School of Public Health, as well as UAB Medicine, as well as the eMERGE collaborators, just generally, as well as just in this paper. So those are the individuals that helped with this paper. Funding through NIH, NHLBI, and NHGRI through the eMERGE network. With that, I will take any questions. I have a few. Hopefully I didn't throw too much genetic information at you first thing in the morning.
Thank you, Dr. Armstrong, for that fantastic start to our symposium. We have time for some questions, so to invite people to come to the front, introduce themselves, and let us know where they're based, and ask their question.
Everyone was blown away.
So maybe I can ask a question while the audience members are thinking about it. What are the potential implications for this study with respect to who might respond to prevention efforts?
Right. So that was kind of the main goal. A lot of heritability estimates, and I kind of mentioned it a little bit before, but didn't really go too far in it. These heritability estimates are really just telling you what the top genetic contribution could be. So say I have a heritability estimate of like 40%. Basically, a polygenic risk score, any sort of genetic risk stratification, would really tell you about 40% at best case scenario of the risk of that specific disease. So in this case, T2D. So when all of these different studies are being generated, and a lot of people don't do heritability studies much anymore. They kind of just jump right into polygenic risk scores and be like, yeah, we know that it's genetic. But you don't really know what that level is. I mean, would you absolutely do it if you had a heritability score of 15%? Maybe, maybe not. I guess it kind of just depends on the disease. But when they're very European-centric and we're just focusing on, we just want, these 2 million people and, in UK Biobank and MVP and, they're not very diverse populations, you're really leaving a lot of people behind in these things. And well, this study, it does, and again, unfortunately, it was just kind of data limiting based on the data that we have locally at UAB, and then we're part of. A lot of our other population sizes were just too small to do other ancestries, but this was really just trying to get out there that yes, these heritability estimates do work. We should be focused on doing ancestry-specific analyses as well as multi-ancestry analyses, and we can't just base everything on sample size. Sometimes it's you do need to do some ancestry things. I don't know. I kind of answered that, right? I didn't go too far around in there. Yes.
Hi, great talk. I'm Mary Ellen Vajravelu from Pittsburgh. I'm a pediatric endocrinologist and seeing increasing amounts of kids with type 2, so really interested in this topic. My general question is about multi-ethnic, multiracial children and how we take these findings and apply them to that, because, this might be my ignorance about genetic studies, but how does it work if you have population ancestry across the world and applying that?
Absolutely. That's a great question. And that really is one of the hardest parts of this whole genetic risk score, gen epi kind of field. So so this was African ancestry specific. A lot of when we're looking at risk score development, and this measurement isn't clinically, so this is population level. This is not individual level. We're not measuring risk. We're just looking at that, again, that upper bound. But these risk scores, we're creating our multi-ancestry kind of for that reason. No one is, 100% anything. So everyone has different allele frequencies, allele patterns, their LD structure is different. And so the field is really moving more towards these multi-ancestry, but making sure that the multi-ancestry is in fact multi-ancestry and not 80, 90% European. Because while, that 15, 20% of other ancestries, when you break that down into the 5, 6 other ancestries, it could be, those numbers are very, very small. And so you're not really getting that genetic architecture that you would hope. So these multi-ancestry summary statistics and analyses are kind of where they're going. So eMERGE has a whole bunch of work looking at multi-ancestry polygenic risk scores, which are kind of more applied and hoping one day that they can, be used clinically. Because that would be helpful with children.
That's fantastic. Thank you, Dr. Armstrong.
Thank you so much. Appreciate it.
The next speaker is Dr. Armando Pena from Indiana University in Bloomington. And as we do this, we're very intentional about the way we identified our speakers, because now Armando is going to talk about interrupting the intergenerational cycle of type 2 diabetes. Awesome.
Thank you, Dr. Degani, and thank you to the ADA for the invite. I'm going to talk really fast here to try to get this done in 17 minutes. Okay, I got no disclosures, and you're welcome to take pictures if you wish. So I'm going to probably breeze through these first slides just because it's the epidemiology that most of us know. So we know that pre-pregnancy obesity is high. Gestational diabetes, or GDM, is also high, and these are data showing from 2019, and you can see that in 2023 the picture has gotten darker. You see some more mustard and orange and a little bit of red, just showing that gestational diabetes is increasing. And this is alarming because these two phenotypes have been associated with type 2 diabetes in the pediatric population. You can see a much stronger much stronger association with gestational diabetes as compared to obesity. So this creates a problem for us, especially because using these data from the SEARCH study, they projected what the type 2 diabetes rates would be in 2060. And you can see that in certain racial ethnic groups, those rates are definitely expected to climb. And this is based on if we were to see the same incidence rate that we see now using those SEARCH data. And so I'm really curious about, what is it in this pregnancy and also the postnatal period that is associated with developing type 2 diabetes, either in childhood or adulthood. So I'm going to breeze through this quickly. I don't want to act like I know that this is a complete causal chain, because it's not. This is coming from many different studies. But just to go over some of what we think is the pathophysiology, we know that obesity is associated with an increase in amino acids, activates the mTOR pathway, is associated with an increase in placental size, which we know leads to an increased fetal weight. But we also know that obesity is associated with some other things like lipotoxicity, oxidative stress, inflammation, and of course there's epigenetics factors that we've been finding more recently that are associated with type 2 diabetes later down the road. And in infancy, we know that obesity is associated with an increased birth weight, increased adiposity, rapid infant weight gain. We see these phenotypes in infants as a result of maternal obesity. And of course, later down the road, we know that obesity is a major risk factor for type 2 diabetes. It will drive insulin resistance, further exacerbate the beta cells, and therefore lead to type 2 diabetes. Again, in childhood, we got inflammation, oxidative stress, hyperinsulinemia, all these things, this physiological milieu that contributes to the development of type 2 diabetes. Now, with gestational diabetes, we also see some of these similar things, right? So I won't rehash those, but the difference with gestational diabetes is, of course, the hyperglycemia, which also leads to hyperinsulinemia, which we've seen in cord blood, thanks to the HAPO study. And interestingly, with GDM, a recent paper came out that showed not rapid infant weight gain, but stunted growth. And those data are right here, and we can see that if we look at fat mass here on the left, this kind of somewhat blue-greenish line is showing a stunted growth in fat mass as compared to those who were not exposed to GDM. You also see it here in body fat percent, and a little bit on the fat-free mass towards the end here. But nonetheless, we see this stunted growth in adiposity. And my main point here, or I do want to acknowledge undernutrition is also associated with low birth weight, which can also increase the risk of childhood obesity through probably different mechanisms. And so there's this catch-up growth that's associated with low birth weights that we think is due to increased adiposity. And so again, my main point here is not to act like I know what's going on here, but really that there's a lot going on here, and there's a lot that we still don't know. We're starting to learn some of these mechanisms that are happening in utero, and we're finding things that are associated, but we still really don't have these causal links. Nonetheless, these are different phenotypes, and do we treat them the same, right? Because they're leading to, it looks like, distinct infancy phenotypes. And so this is, bear with me here, it's a very basic science study, but they took islets from people with normal glycemia, and they dipped them in vitro either in glucotoxicity, lipotoxicity, or glucolipotoxicity. And so you see that in each of these conditions, the GSIS is glucose-stimulated insulin secretion. So all of them exposed to these conditions demonstrated perturbed insulin secretion. However, only the glucolipotoxic conditions demonstrated cell death, so beta cell death. And the interesting part is they looked at some epigenetics markers, and you can see that as you travel down the line here from glucotoxicity to lipo to glucolipotoxicity, you can see that there's an increase in the DNA methylations of certain genes. So there's much more going on as you move down this line, and this goes back to the last slide of, if we consider this, again, this is all in vitro, so take it with a grain of salt, but if we consider this glucolipotoxic as our GDM with obesity condition, are we supposed to treat this the same way than just obesity alone, right? And is targeting the hyperglycemia, if we think back to the last slide, is that enough, right? Is that also improving some of these other mechanisms or things like amino acids that we think are contributing to the increased risk in the offspring? So we see the guidelines here for obesity and gestational diabetes during pregnancy, and we know that it's lifestyle intervention, whether you're looking at ACOG or ACSM, there's slightly different recommendations, but nonetheless, it's lifestyle intervention, right? Do some physical activity, balance nutrition, and ACOG would say that in the second and third trimesters that you need an extra 300 extra calories per day. In terms of the ADA, they say that with GDM, also lifestyle intervention, and then if you need insulin, to apply that as needed. Now, I know that at one of our health systems in Indianapolis, these two phenotypes might dictate a different pathway to treatment, right? And so we know that individuals with GDM, they're referred directly within the EHR system to medical nutrition therapy. So that's an intensive session that you go through with a registered dietitian. But with obesity, right, if you do your OGTT, you don't get, you're not diagnosed with gestational diabetes. We're just, maybe you get a pamphlet and we say, hey, you got to do some exercise, just stay healthy and wish for the best, right? These are two different phenotypes. We know that they're both associated with type 2 diabetes, and so, should we be looking at things here within our own health systems to make sure that we're addressing these issues appropriately? So here we see that prenatal lifestyle intervention in women with GDM is associated with large for gestational age. Not too many studies here, but you do see a relationship that there's a reduced risk of LGA. Here with maternal glucose, again, not too many studies, but you can see that in the middle here, these are looking at the postprandial glucose markers, so probably from OGTT. Much greater effect here in terms of improving glucose tolerance. A little bit of movement in fasting glucose, but not significant, and very little movement with HbA1c, which we would probably suspect within a 9-month period. And so going back to this picture, what about these things, right? I think this is where the literature is lacking, because we're barely starting to find these signals and these associations with some of these markers, because we have some pretty cool technologies now to be able to do this stuff. And so I will say there's not too many randomized controlled trials in this area, but I did find a study that's pretty interesting. And they looked at, it was a randomized controlled trial where they did exercise and they measured metabolism within mesenchymal stem cells. And so these first two figures are looking at glucose oxidation and non-oxidized glucose. Almost found significant effects, but you can see that they're not less than the 0.05 value. We do see that with glucose partitioning, there was a significant effect whereby aerobic exercise in these moms led to, increased glucose partitioning in these mesenchymal stem cells as compared to the control groups. So nonetheless, this is evidence to suggest that what mom does in pregnancy does affect the phenotype, at least as we're measuring it by mesenchymal stem cells. And so what if we missed this whole point here, right? What if mom doesn't show up until delivery? We just missed out on all these things that could potentially be driving the risk of type 2 diabetes. And we know that formula is also associated with childhood obesity and type 2 diabetes. On the flip side, we know that breastfeeding has been associated with a reduced risk of type 2 diabetes, and these come from a pretty good amount of studies, but I would say they're largely observational, which is the limitation, of course. Here, one of my colleagues, Dr. Emily Nagel from the University of Minnesota, demonstrated that the milk in mothers with GDM who were exclusively breastfeeding for at least a month demonstrated these significantly differentiated metabolites, and we went on to find that some of those metabolites were associated with, you might not be able to see it too well, but these are all changes in either infant growth outcomes or infant body composition outcomes as measured by either PPOD or DEXA. But what we find here is that there are some metabolites that are associated with changes in infant body composition. And this is coming from metabolites that are differentiated by GDM in breast milk. Another milk study by another colleague, Dr. Cheng Lu from Boston Children's Hospital, showed an association of all these metabolites with either acute exercise or chronic exercise. So these metabolites were, these people went through an exercise protocol and they measured the metabolites in response to this acute protocol. And then they looked at the chronic exercise through a survey, I believe it was Recall, and they found a whole nother list of metabolites that were significantly associated with chronic exercise. And then they combined the two to see if there were any overlapping metabolites. And what they're doing here is, again, seeing if they're associated with any infant body composition outcomes or infant growth outcomes, and we can see that there are. Now, again, these are observational studies. They're exciting studies, and potentially these are signals that we can start targeting to see if they're actually causal within the pathways to developing type 2 diabetes. Unfortunately, in the US, we have very low exclusive breastfeeding rates at 6 months. The AAP, CDC, and the WHO all recommend 6 months of exclusive breastfeeding, nothing else, not even water, no tea, no soda, nothing, just breast milk. And we do that very poorly. You can see this broken down by racial, ethnic group. This red line is the Healthy People 2020 goal, which is 25.5%. And what's worse is that in 2030, we got to double that. So we got to jump up to 42.4%. And this was back in 2015 on this study, but I think the recent data shows that we're at 27.2%. So we're in 2025, we got 5 more years, and we got to jump up this much. And this study actually did that analysis, and I'm looking at Indiana here because that's where I'm at. We have 97% of an increase that we need to get to that goal of exclusive breastfeeding at 6 months. And across the country, the range is 16 to 207%. So I'm an assistant professor. I just got my job. I'm 6 months into the job, and I'm trying to figure out 20 to 30 years from now when I give another talk, hopefully, are we going to put a dent into type 2 diabetes, right? And how do we get there faster? And how do we make this process during the perinatal period, which is already chaotic? If everything was perfect, it's still chaotic. How do we make this better for moms to get through this period, but also reduce the risk of type 2 diabetes? And so that's kind of what I want to focus on. And I come from a department of health and wellness design is what it's called. We're really focused on human factors and implementation science. And so we really think about the interventions that we design. And when we look at these disparities, I only put the Latino group here, but you can imagine there's other minority groups that are also facing disparities. You know, we believe that this disparity is due to social unmet needs, structural drivers, right? And so we need to address those things. The National Academies charged us in 2019 with integrating social care with medical care. And so ever since, health systems have started to assess for these social needs through their screenings, right, which is really cool. So if we're just looking at food insecurity here and somebody says yes to that in the health system, then they get referred to, at least at this health system, a certain amount of services, okay? So they're either going to some clinical services, social care services, or health equity zone. These are health equity zones in Indianapolis are certain zones that this health system is focused on to because they face really bad disparities. The whole point here is that then you get referred to all these other things, right? So imagine walking out of the clinic and they say, you need to go do this. Here's a food voucher with a QR code. You can go cash out at the Fresh For You market at this health system, which is pretty cool. They got a grocery store that comes in-house. But there's all these things, and this system is very complex to navigate. So imagine if you don't know English. In Indianapolis, we have a really high surge of Haitian Creole population, that it's really hard to address these issues. And so this is just in the clinic, when you're in one appointment and out. And if we're thinking of other systems, right, in human factors, we think of the patient work, right? Once the patient leaves the clinic, what type of work do they have to do to make sure they meet your recommendations, to go get the medicines that they need, or to go get the healthy food that you're telling them to eat, or to get the exercise that they need? There's so many other systems involved that are very difficult for a Latino that doesn't speak English, or a person who's Haitian Creole that doesn't speak English. And so again, these systems are very complex. They don't talk in terms of their databases, and that makes it really hard for us to follow patients to see if they got to where they need to go. I'm going to get really fast here because it looks like I'm kind of running up on time, but we gathered a coalition. We developed a framework. We adapted the NIMHD research framework specific to our local context, and this is what it looks like. This was Priscilla Vasquez who developed this image for us, and we're really interested in this system. So we gathered. Over the last couple of years, we've been engaging these community members or these community organizations to develop a real-world intervention that we can test that integrates lifestyle and breastfeeding, and kind of spans this whole perinatal period to really try to help moms navigate these systems. And really, we're interested. First, we're going to be going to these moms to ask questions about their relationships with these people, right? The people that they interact with, the lactation consultants, the OBs. And then everybody else that's involved in this system. This isn't even everybody. This is just the people that I could think of that I've interacted with within these systems. But it can get real complex. And one of the most important things that we want to focus on is the family, right? We're asking questions like, how does your family, or who do you anticipate to be helping you when you feed your child, right? And then we ask them when they're actually in the postpartum phase, who's actually helping you, right? And that type of information is going to help us build an intervention. So we haven't launched this, but we're getting ready to. We're interested in journey mapping, and so we're following this perinatal journey among women to really figure out what those determinants of physical activity, nutrition, and breastfeeding are. So we're doing some interviews, collecting some cord blood, breast milk, body composition measures, and we really want to help women navigate this process smoothly. And so these little blue dots are some of our ideas, like a community navigator that can stay with the mom very intimately throughout this process, and not just like, hey, see you in 6 weeks, and then never really contact them again. So again, this is very local. The contexts are very local to us, but I think that the processes that we use are what are scalable. And so we're really using community organizations that are already scaled. Right? So if we're trying to influence population level outcomes, how do we get there quicker? And I think we have a lot of systems that are already there, although they're kind of under attack, some of these. But nonetheless, they're already there. We can leverage them, and we should leverage them, and we should test it locally so that if it works, then we can scale it and try to test it in other areas. So this is our implementation framework. We're using the EPIS framework. We've explored a lot. We've done gauging of the community. We got together to build our own framework that would guide our research. And now we're in this yellow phase where we're doing that one study. We're doing journey mapping to understand what these moms are going through from their perspectives. And the goal is to get to implementation by doing some hybrid trials. I think we need innovative interventions. We need more efficacy trials on novel ideas, right? Are there other biologics in pregnancy that we can use? Can we target insulin sensitivity directly instead of just treating the hyperglycemia without affecting the baby? And measuring novel biomarkers like some of the things, amino acids, lipids, and such. And then I think that we need to integrate human factors engineering with dissemination and implementation science to develop interventions that are actually scalable and to disseminate those processes so other people can replicate them elsewhere. And I just want to thank the village that helps me in all this work. And really most of these people are mentors that are amazing folks that have helped me along the way. Thank you.
Dr. Pena, that was fantastic, and thanks for taking us all the way through biology to the clinical workflow to implementation science. We have time for some questions, so we'll ask audience members to come up, introduce themselves, and tell us where they work.
Great talk. I recently completed my BS, and I'm an incoming PhD student in molecular biology.
Awesome.
I was
Congrats.
interested in your in vitro islet study because my lab also exposes these islets to high glucose and then looks at ways of preventing that perturbed GSIS through beta cell rest. And I what caught my eye was, that marked increase in DNA methylation when you have the high glucose and the fatty acids. So I'm wondering if it would behoove basic science labs like my own to look at that intersection between glucose and fatty acids more to see some of those differences in markers and phenotypes like you mentioned.
Yeah, congrats, first of all. And I will say that study was, I think it was a review of 3 isolated studies, I think from the same lab that looked at that. And it's the same author. And I'm just going to say that is not my area. So I'm not even going to act like I know how to answer that question. But obviously it's interesting, right? If you're exposing some beta cells, which is what they did to these different, either glucose or lipids, and you're finding different signatures, and you can see clearly that there's more DNA methylations and then more on when you combine the two. That's interesting, right? And it makes you think. It makes me think about treatment, right? If we're just telling everybody, hey, here's a slip, get some exercise in and do some nutrition, and we just leave it at that, that person's probably going home and not. I used to be a personal trainer, right? I used to make programs for people, but I also used to train them in person. That's much more effective when I'm there versus, hey, here's a program, good luck. I know they don't do it, and I know they didn't do it. But yeah, sorry, I don't really have a lot of insight to the very specific mechanisms on the epigenetics part.
No, but I think it's translatable just in the idea that, these discrete sort of methods don't always work when we have such a continuous population. So thank you
Gotcha.
for your
Thank you.
talk.
This will be our last question.
Right. Juliana Camargo from NIH and HLBI. I want to congratulate you for the amazing work that takes a village and a lot of work.
Thank you.
I would like to ask you if you can expand a little bit on your framework for implementation. You mentioned about trying to leverage all these programs we already have available, but I would like to hear how you're incorporating the views of the patients who face the challenge about being, to manage gestational diabetes with all the challenges of being a new mother. So could you expand a little bit on that? Thank you.
Sure. Great question. Thank you. That's obviously important to have their perspective. I started as a postdoc at IU 2.5 years ago, maybe. My first job was just to get into the community and see if this is an issue. I was really focused on the breastfeeding aspects of things because I was interested in the data that shows it's not only helpful for the baby, but also for the mom in terms of reducing diabetes risk. So we gauged partners, right? I went from meeting to meeting and I followed up on leads, right? Just having coffee with somebody from WIC. And then they said, hey, you need to meet this person. And I went to meet that person. So that took a while. That was about a year maybe just to formulate a group that was interested in this. Then we developed a coalition. I said, okay, well, let's make this a little more rigorous. So we gathered together in working groups. We were all on the call, and these are all professionals. There was one community member who's like really a community advocate for breastfeeding. This next step, the journey mapping, that's going to get the information from the patients. So we really haven't gone to the patients yet. This is like 2 years into just gauging the community and seeing if there's an issue here and seeing who the main players are to leverage for an intervention. Now I want to go to the patients to say, what do you guys say? I heard what the professionals said, but what do you guys say? And what are the issues around you guys? Because it might not be anything related to diabetes or a health behavior. It might be like, I'm scared to walk outside because there's crime out there. So yeah, we're getting there, and hopefully in the next couple of years, I'll have some more data around that.
Thank you, Dr. Pena.
Thank you.
As we move from heritability to interrupting the intergenerational cycle, our next speaker is going to talk about preventing type 2 diabetes in high-risk families. Dr. Lire Rascon is a clinical associate professor at Arizona State University.
Good morning. Thanks for joining me today. And thank you to the ADA and Dr. Dugani for inviting me to speak today. I'm Lidia Resco, and I come to you from Arizona, where I am still in the Arizona time zone, so this is especially early for me. I have not adjusted yet. I'm going to be sharing with you a little bit about preventing type 2 diabetes in high-risk families, focusing specifically on Latino families. I have no conflicts of interest, and you may take photos. So my prior speakers have really teed me up nicely in terms of my background here. We're now very well aware of the heritability of type 2 diabetes and that Latinos are disproportionately impacted by type 2 diabetes. One thing that's interesting, though, when we look at the high risk of type 2 diabetes among Latinos, Latino children have the highest lifetime risk for developing type 2 diabetes. And I'm fortunate enough to cite my colleague here, Dr. Pena, in some of the work in adapting the diabetes prevention program for Latino youth. And as this work was done, and as we're more aware of heritability and how these diabetes prevention behaviors occur for a youth, for Latino youth, it happens in a family system, and it happens in that context of the family. And so the next question comes, what would happen if we were to adapt now the DPP for the family system, which our team has done, and we're currently in a randomized controlled trial where we're evaluating the efficacy and implementation of a family-based diabetes prevention program. So I'm going to share with you just very briefly about this family-based diabetes prevention program. It's a 4-month diabetes prevention program, and while it is based in the NDPP, the CDC-recognized curriculum, there are a lot more contextual factors that have been implemented to create this family-based diabetes prevention program, from existing evidence to stakeholder interviews and focus groups, community advisory boards, and pilot studies. And it's also based on this conceptual model you can see here, where we look at unifying families as a health-oriented system that's focused on increasing health behaviors. So you can see these family processes at the bottom: resilience, empowerment, engagement, and cohesion, working through these skills and behaviors, so things like increasing physical activity, improved eating behaviors, with the goal to improve health outcomes, and in this case, reducing type 2 diabetes risk and increasing quality of life. So I recognize that these family processes, resilience, cohesion, empowerment, and engagement, carry a lot of connotation, sometimes controversy, and so I want to share some very clear definitions that we've created for this specific conceptual model in the context of diabetes prevention in families. So engagement has been defined as interacting as a family with other families, with health educators, and the environment to take actions to improve the health of the family system. Empowerment relates to acquiring knowledge, skills, capacity, and that's to identify and utilize resources to improve health and reduce diabetes risk. Resilience relates to leveraging strengths, relationships, cultural values, and assets in order to respond to pathogenic forces that underpin type 2 diabetes, again, so the family can flourish as a healthy system. And then cohesion relates to those bonds that the family has within the family to decrease conflict, prioritize health of the family system around a shared purpose health goal. So when we look at adapting diabetes prevention for the family, it gets very complicated. We have to not just think about a person, we now have to think about the family system and the family processes. So thus emerges my research question: how do family structures and processes influence type 2 diabetes prevention in the family system? So to do this, I conducted a qualitative descriptive study. We did semi-structured interviews with families in the intervention group of this randomized control trial, the FDPP. We're currently in cohort 10 of 10, so still ongoing. We learned in a pilot stage that doing one interview with a family is very messy. You have children correcting parents, you have children not wanting to say things in front of their parents, you have parents, it's not quite as valid as if you were to do individual interviews with the youth and interviews with the adults. So per family, we did 2 interviews, one with youth, one with adults, sometimes 5 youth in an interview, sometimes 1. Again, with the adults, sometimes 2 or 3 adults, sometimes 1. Interviews were conducted in English or Spanish, depending on the preference of the family. So all of the youth actually preferred English. Most of the adults preferred Spanish. And then we defined family structures as the household members in relationship to the FDPP the RCT participants. So to be in the RCT for the Family Diabetes Prevention Program, the inclusion criteria required one youth, aged 10 to 16, with a BMI above the 95th percentile, and an adult, that is either their parent or caregiver, and then everybody else in the household was invited to participate in the program. So we described the household structure and the family structure based off of those participants. To analyze our data, we did line-by-line content analysis in the source language. We were fortunate enough to have a bilingual, bicultural research team. So for example, if I conduct an interview in Spanish, we analyze the data in Spanish. We didn't translate it and then analyze the data. And anyone in here that does research in multiple languages, you know, and as you can imagine, whether or not you do research in multiple languages, the second you translate something, you lose a little bit, and then you lose a little bit more as you translate it. So we didn't translate any of our data until the very last stage where we're presenting our data. We conducted inductive and deductive coding. So we created codes based on those 4 tree family processes, and then also the family diabetes prevention curriculum. And then also certain inductive codes started to emerge as families were sharing their experience that didn't really fit in any of those boxes per se. So sharing the results, just the families that we've interviewed 18 families to date. 18 times 2 is 36. So we have done 36 interviews, one youth, one adult family, or one youth, one adult interview per family, with a total of 33 youth participants in those 18 youth interviews and a total of 25 adult participants in those 18 adult interviews. Just sharing a little bit of demographics for the interviewees here. The average household size was 5.7. You can see that the adults predominantly preferred Spanish and were from Mexico, so born in Mexico. The remaining participants were either from Honduras or from the United States. And for families with parents born outside of the United States, their average number of years in the United States was about 18.1. And I've shared some attendance rates here just to provide a little bit of context. So the Family Diabetes Prevention Program has 2 classes a week. On one day, it's a nutrition class. On the other day, it's a physical activity class. And so I wanted to share participation rates here. So you can see nutrition, our rates range from 33 to 100%. And same with physical activity, 26 to 100%. So sharing that we interviewed families that were self-proclaimed bad students, and we interviewed families that went to every single class to really show that range of perspectives. For family structure of these 18 families, they were predominantly 2 parents and biological children. You can see 8 of the families there. And then we also had 6 families that were a parent or parents, their biological child, and then any extended family. So this may look like a cousin, this may look like a parent's sibling, so an aunt or uncle. It may look like a grandparent or grandparents. It may be 2 sets of grandparents, so any extended family. And then single parent and biological children, we had 4 families, and those were all actually single mother families. So when I share with you the qualitative data, if we just had about 6 more hours, I could go through all of these codes with you, share all of these wonderful stories that these families shared and all of the sub-themes under each of these. But I'm going to just give you kind of a broad overview of what we're seeing from this data so far. So in the middle, you can see, I do have a pointer here. I won't struggle with my technology for a second. In the blue, you can see family structure and diabetes prevention. So we looked at how different family structures and the roles within the family shaped specific components of the Family Diabetes Prevention Program. And you see kind of a rogue theme here, convivir, it says in brackets, living together. And that is one of our inductive themes that kept emerging. And if anybody in here speaks Spanish, when you translate convivir, it translates to co-live or cohabitate or living together. And that still doesn't capture what convivir really means. And it's this really genuine being with one's family and one's people. And so this was something that really strongly emerged in the data. And then what we did for our remaining themes was take those 4 family processes at the base of our tree and look at how those shaped barriers to type 2 diabetes prevention and how those shaped facilitators to type 2 diabetes prevention. And simple math tells you we only have 3 under each. And the reason for that is resilience, our 4th family process, wasn't truly just a barrier and wasn't truly just a facilitator. When families spoke about their resilience in type 2 diabetes prevention, they spoke of the barriers that they faced and then leveraging their assets and their skills and their knowledge and their resources to overcome those barriers. So that's why it's centrally located. So what I'm going to do for you today with the next 10 minutes I have is go over 2 themes that kind of really illuminate some of the family structure themes that we saw, as well as really captures what these families went through in the diabetes prevention program. So when we look at cohesion and cohesion barriers, this was really describing how issues with cohesion or lack of cohesion created barriers to type 2 diabetes management. And so our sub-themes here were family divergence and family commitment to lifestyle change, which described how some members were very committed to diabetes prevention behaviors and activities, and others just either were not motivated, were not able to be involved, were not interested in being involved. It was not always this personal trait. It sometimes was physically unable to participate. Challenges with children presented as different issues with not wanting to eat healthier foods, maybe not wanting to engage in exercise, but also just the complexity of children's lives now and being busy in school and being exhausted from school and having stress coming from their school and their social lives. And then family schedule. I interviewed many families where dad gets up at 3 and comes home at 3 a.m. and comes home at 9 p.m., is not eating with the family, is not able to eat with the family, or grandma and grandpa live upstairs and we don't really see them a lot. And so therefore the decisions we make and the conversations we have are fragmented because of our different schedules or our different structures. And then communication and conflict in the family described how different families would communicate differently, that maybe they were very together, but they had a lot of conflict when they were together, or they were having communication issues. So cohesion was also a facilitator. So some of our major themes that surfaced here: cohesion perspectives and health of the family. Many, many times participants would share, this is not about my health. This is not about me preventing diabetes. It's about diabetes prevention in my family and my family system. And it wasn't just moms and dads saying, I'm doing this for my kids. It was kids saying, I'm doing this for my mom. And they would tell me, I'm so proud of my mom. And if you could sit in an interview with a 15-year-old boy saying he's proud of his mom, it's pretty magical. So we heard kids talking about and parents talking about cohesion activities and talking about elements of the diabetes prevention program where they would not just go and exercise by themselves, but they'd be exercising with their family. And nutritional activities, buying food together, preparing food together, eating together. Behavioral health, talking about stress and coping as a family, having family meetings, and problem solving, which had a lot of similarity with resilience as well. So I'm going to pick at two specific family structural, interesting things that arose with family structure and type 2 diabetes prevention in this study. One with families with adult extended family. So as you'll remember, we had 6 families who were a parent and a child, or 2 parents and a child, and then maybe an aunt, a cousin, or a grandparent. So when that other family member was an adult, some certain things started to surface. And then I'm also going to touch on roles and influence of children. So when you look at households with adult extended family, this did present as a barrier. One of our themes, that family divergence and commitment to change. We found that families with an adult extended family member, about 5% of their codes were related to family divergence, as compared to other family structures was only 2% of codes. And so as you can imagine, we have more adults in the household having their own individual behaviors and more opportunity, in a sense, for them to not be committed to type 2 diabetes prevention. So I'm going to share a couple of quotes here that relate to this. So this is a family that had multiple adults in the household. Each person has their activities, their expenses. We all live in the same house, but each person has their own life. And then here we have a grandmother that lives with participants. Yeah, we would tell her, our grandma, about the program, but she'll be like, I don't care, I'm going to get my Starbucks. She loves Starbucks. And then I asked what she ordered at Starbucks, and it was not black coffee. And then we have another family that has multiple adults in the household. It's complicated because we live with 2 other people who aren't part of our life plan. So sometimes we're really spending time with them in the evenings and they don't have the same intentions as we do. It makes our relationship a little difficult because if we want to, say, avoid soda, which everyone loves, well, they don't have that intention and they buy soda, and then my son sees the soda and wants it. So yes, it becomes a bit complicated when not everyone in your household shares the same intention or goal. But having an extended family member was also a positive and was a facilitator. It was often seen as a resource. We have more income now, more adults can work. We have more support, we bring more skills to the table. So we saw a lot more coding of cohesion perspectives and this perspective of health of the entire family as well as activities. So this is an adult that lives with her sister and her child. My sister who lives with us, she tells me, you need to walk, you need to go to the gym. So we have adults kind of inspiring each other to engage in diabetes prevention. And then this is a child telling me she came home without her parents at home, but her grandmother was at home. It was already getting dark, so instead of just walking to the park, we walked around the block with my grandma. So they were able to engage in physical activity because their grandma was home. And then this is another family with multiple adults, and the other people we live with, sometimes they invite us somewhere to swim, to go to the lake, to go to the park. So when we look at children in the family system, there were quite a few codes about challenges with children and family divergence. You can see here 7% of the codes related to family divergence were in single mother households compared to less than 1 to 3% for other families. And it's not just about children don't like to eat their vegetables, although as a mother of a 2 and a 4-year-old, a lot of kids don't like to eat their vegetables and it is a big challenge. But that wasn't the only thing that emerged. Sometimes these kids were stressed at school and sometimes they didn't want to engage in physical activity. So we have a single mom here sharing that sometimes the kids win because we don't want to tell them no to soda, but we're actually harming them. So it really created that awareness of, I know I need to quote unquote make my kids engage in this healthy behavior, but I'm exhausted, or sometimes I'm just going to quote unquote let them win. And then here we have, my son told me he didn't want to eat the food I made, and I told him, it's you who asked me to buy hamburgers, it's you who asked me to buy pizza, and I said, sometimes I buy it and you don't want to eat anything else. When I cook at home, you don't want to eat it, so I'm not forcing to eat the pizza, yet you do. And this is why we did separate family interviews. You can really sense the tension here, and these parents were very frustrated because they knew that the risk that their children were under, and yet they were struggling with helping their kids make these choices. And this next quote is actually from a youth participant. Sometimes I go buy chips without her knowing, and then I hide them under the covers. And so that it made me chuckle, and then I was like, oh, these poor parents. This is very stressful for them, and yet these kids are also stressed, and they feel the sense that they need to hide this because they also know that they need to make these changes. Children are also helpful. Children also facilitate type 2 diabetes prevention. So this explaining some of the cohesion activities, kids helped their parents be more active and the adults be more active and each other, as well as this perspective of health of the family. So here we have a dad saying, he says to me, hey dad, let's exercise. And well, so I don't let him down, like we say, don't leave him hanging. I say, okay, even if I'm tired, we start exercising. And then we have here my oldest sister and well, almost the whole family were diabetic. My grandmother died of diabetes. Both grandmothers died of diabetes. My oldest sister says, take care of yourself, take care of yourself. That child is so young. I told her when she came to visit, she was really excited because she saw us exercising and she said, oh, at least you're doing something. So sometimes it extended outside the household, and this awareness of families wanting to prevent diabetes throughout and wanting to do quote unquote something. Then here we have a single mom. This quote is just exhausting for me to even read. So sometimes it was a bit harder for me because I had to go to work, then come back and cook, then go pick them up from school or whatever, then go to appointments and then get back here and feed them and then go again. Sometimes it was really complicated, but I still went to the FDPP for them. And so we had these exhausted parents working long, long hours and still pushing their kids to engage in diabetes prevention because that was their motivation. So I alluded to convivir, this concept of really genuinely being with family and being with each other. And I wanted to share just a couple of quotes here that really leverage how family-based interventions can really accelerate diabetes prevention. And so one family mentioned, if we communicate, we're able to keep going, but if not, someone's going to be left behind. And that's why we all stick together, so we can go back, lift them up, and bring them with us. And another family mentioned, we always eat together all the time, never someone separate from the others. No, always, everyone together all at once. And so really talking about how I think when we look at diabetes prevention, a lot of times we are encountered with that individual and we see that A1C that's elevated, or we see these risk factors and we think about their tailored plan. But a lot of times we don't think about where they're going home to and how that's going to influence their behaviors that they so desperately want to change, but are much more challenging to change if we don't consider those factors. And so that leads me to my conclusion and my take-home points for you, that family structure and processes are integrated into diabetes prevention behaviors, and that going forward, diabetes prevention interventions really should include an assessment of household and family structure, family assets and motivators, and then build upon and incorporate family structures and roles and motivators and assets. This really brings to light some of my prior work with grandmothers who were managing diabetes while caring for grandchildren. And I would speak with one grandmother who was caring for 5 grandchildren and would say, I don't have time to check my blood sugar. I don't have time to exercise. I'm not able to manage my diabetes. And another grandmother who was caring for 5 grandchildren had very similar barriers and said, I don't have time to go to the gym, but 8-year-olds love to dance salsa. And 8-year-olds are also very good at reminding you to do things. And so kind of leveraging, what are your roles? What do you have? How can we look at your family structure and your family processes as really assets for diabetes prevention? So I just want to acknowledge PI on the RO1 that was funded by the NIDDK for this grant, Dr. Gabriel Scheibe, and my late colleague Allison Williams, who was my co-coder and partner in crime for this work. Thank you.
Thank you for that fantastic presentation. We have time for one question. So whoever wants to come to microphone first gets it. What's your name and where you're from?
Hi, I'm Dr. Alexandra Moran. I'm from University of Maryland, and I work a lot with the Latino community. One of the things that we saw was that involving parents and kids would tell on their parents when they were not doing. We would partner with restaurants to teach them how to try different foods, still Latino food, but different origins. One of the things I struggled the most, and I don't remember if you showed that, was the recruitment piece and the retention piece, because I would offer free A1C screenings, free everything. They would come get tested, enroll, attend the first class, and then slowly trickle down, which it happens in DPP, the year-long, the CDC one. We did a 3-month version one, and it was just very hard to retain the family. So,
if you could answer that question for me. No, in our current RCT, that's exactly what we're facing. Is our retention rates have been a really big struggle, and I think retention rates, regardless, as you mentioned in the DPP, are challenging anyway with an individual. When you bring in a family and all of the factors of every family member and all of the barriers with a lot of these families, it is a clear barrier. We offer financial incentives. We even offered Uber or Lyft, so sometimes transportation is a barrier. So looking at what are the barriers that we can actually do something about, but then it comes to a point of wasted resources where we've reserved lab time and we have all these staff that are being paid and we have all of these no-shows. And so that is something that we are really trying to confront now. And some of my other work is in, social needs and trying to understand what are their unmet needs that are much bigger priority than coming to the lab and getting their labs drawn and showing up for classes. And I think a lot of times it's a lot of touch points, a lot of contact, having either community health workers or staff members staying in touch with them, checking in with them. Families have shared that I feel really comfortable talking to your staff and I feel like we're friends. And so I feel like if I don't show up, I'm kind of letting them down. But creating that at a large scale is very challenging to do. So I wish I had a good answer for you. But thank you. Good question.
Thank you, Dr. Nestor.
Thank you.
Could you just...
Yeah.
If not, our speakers may be able to stick around for a few minutes, but otherwise, thank you all for joining this symposium and for all the interaction. Thank you.